So now I'll hand over to Dr. Gabriela, who will curate the next session.
Good morning. I'm Gabriela Repetto from Clínica Alemana, Universidad, Desarrollo in Santiago, Chile, and happy to moderate this session on genomic diversity that, as you know, encompasses the vast array of genetic variation within human population and has significant implications for research, healthcare, and society. This session aims to explore the rich tapestry of genomic diversity and highlight its role in shaping individual traits and susceptibility to diseases. I'll remind you to submit your questions through the Pigeonhole app, and you can scan the QR code that you see on the screen. And now I'd like to introduce our first speaker, Dr. Biju Viswanath, who is a distinguished psychiatrist and researcher currently serving as professor of psychiatry at the National Institute of Mental Health and Neurosciences in Bangalore, India. His expertise lies in the molecular basis of etiology and treatment response in neuropsychiatric disorders with a specific focus on obsessive-compulsive disorder.
Good afternoon. Sorry, good morning. Sorry, it's a different time in India. Okay, so I'm very happy to be following Professor Vajira because I think a lot of the, so I come with a disclaimer. I work in a low middle income country with low resources, and a lot of the things that he told about what difficulties we face, it was heartening to hear because it is a struggle to do work, and we have tried to do some work, so I'll be trying to show some glimpses of the work that we have been doing in India. I want to keep it this way. Oh. Sorry. Okay. So I work at the National Institute of Mental Health and Neurosciences. I'm a psychiatrist who got interested in genetics and stem cells after that. So I'll show you some of that work. So this is the National Institute of Mental Health and Neurosciences. We are in Bangalore. Bangalore, you can see as a red dot in the south of India. So it is the largest clinical teaching and research center for diseases of the brain and mind. Essentially, we only cater to psychiatry, neurology, neurosurgery, and child and adolescent psychiatry. These are the only clinical departments we have. But we have a 1,000-bedded hospital. We have academic faculty doing a lot of research with collaborations, a lot of molecular work that we try to do. Yeah, but it's a wonderful campus. People should visit. So why mental health? So 1 in 7 Indians, of course, 1 in 7 people in the world, is affected by a mental disorder. And this accounts to 15% of total years lived with disability. A lot of these illnesses are well known: depression, anxiety, psychosis, childhood developmental disorders. Dementia-related burden, we are all getting older, is expected to increase. So it's not just about the mortality, the morbidity, people commit suicide, people die with medical causes untreated, which is very common in India. Stigma is a huge problem. People don't know why the illness happens. So when I started my research, I thought, okay, let me try to understand how the illness happens, why it happens, and then that would probably help. So we know that almost all psychiatric, the onset of many psychiatric illnesses is in the prime of life, early adulthood, adolescence. Even before they're diagnosed, many of them have childhood problems. So a lot of these conditions are neurodevelopmental in origin, not just early neurodevelopment, but late neurodevelopment. So we know from earlier research from the group that if you look at MRIs of children, of patients with mental illness, in this case children with alcohol use disorder or externalizing disorders, we find brain changes. Now this is seen in 50 years before the onset of even neurodegenerative conditions. This is scans of patients with the clustering mutation for Alzheimer's, indicating that these changes are happening in these brains way before the onset of symptoms, and that is when we start seeing them in the clinic. So we need to catch them before that. So we know this, that of course genetics plays a huge role in getting people to a risk state to develop illnesses, but of course in psychiatry, in mental health, there are prenatal environmental factors, postnatal environmental factors, all of them contribute. And at some point, they develop all these conditions: schizophrenia, bipolar disorder, autism spectrum, ADHD, OCD, major depression, all kinds of illnesses which come up at some point of life. So what we tried to do was start a program. This is in 2015. This is government funded and now funded by philanthropy. So we are trying to capture multiple affected families because we thought that is where we could find the biology in a much better manner. So we were looking at multiple affected families with psychiatric illnesses, prospectively sample them, follow them up, do longitudinal assessments, make cell models, try to understand what changes are happening. And this is what we have been doing for the last 7 to 8 years. So I'll show you snapshots of this work. It's very broad, and I'm happy to take questions later. So this is the length and breadth of India, although we are in South India. So we have been able to capture patients from all throughout the country because it's a tertiary referral hospital. So 750 families, each with 2, 3, 4, 5 people affected with severe mental illness. Around 3,000 individuals. We've been doing 2 yearly follow-ups. More than half of them with MRI scans, psychophysics, neuropsychology. We didn't have the funds to do the sequencing for all of them, so we have done for a subset. We have kept lymphoblastoid cell lines, lymphocytes, plasma. We have generated pluripotent stem cell lines for a subset. Of course, we have a lab archive of 30,000 samples in the last many, many years. So these are the kinds of assessments that these people go through. I mean, people are very cooperative. The research participants are extremely cooperative, and we should be thankful to them. So they go through a detailed assessment for 13 hours, 10 hours doing a lot of these things. Of course, genetics, DNA. We do assessments of physical anomalies, temperament, personality, behavioral endophenotypes, the neuronal endophenotypes, or the neuropsychology, MRI, FNARS, EEG, eye tracking, environmental risks, the socioeconomic vulnerability, the adverse life events, adverse childhood life events, recent life events. Of course, a lot of assessments about the phenotype, the severity, the treatment response. It's a lot of time that these people spend with us. So I'll go through some of these aspects. Talking about phenotype, just one thing. We find that many of these, we spoke about pleiotropy. So we know here that if you look at this pedigree, I don't have a pointer, sorry. There are people with bipolar disorder, psychosis, epilepsy, alcohol dependence, multiple sclerosis, all in the same family, indicating a great range of conditions which are happening in this particular family. We have a lot of families like this. If you look at panel B, these are in the x-axis, the diagnosis, in the y-axis, the number of families. So you can see all kinds of combinations happening in these families. Looking at environmental risk, so we looked at adverse childhood experiences. Around 2/3 of these people had adverse childhood experiences. That's a lot, 2/3 of the whole sample. Now, what we also find, interestingly, if you look at panel B, this risk seems to be more in men. So although both men and women are exposed to adverse childhood experiences, the effect of these experiences in terms of having the psychiatric illness earlier or having greater severity of psychiatric illness seems to be more in men. This is again something we are exploring to understand why this is happening, but that's a question. Cognition or neuropsych. Now, as you can see in panel A, so these are the various factors in the x-axis, the general factor cognition, new learning and recall, social cognition, working memory, and cognitive flexibility. And if you look at the black dots, the general factor cognition is abnormal across diagnosis, indicating that it is transdiagnostic. Of course, there are other factors also which are transdiagnostic. Now, this is in affected members. Let's look at unaffected members. If you look at unaffected members, again, general factor cognition is abnormal transdiagnostically in the unaffected members of these families as well. That also indicates that these people are more vulnerable to develop these illnesses in time. So then we try to understand, okay, what is the cumulative effect of adverse childhood experiences and cognition? So herein, we try to look at this. So if you look at panel A, we are looking at the effect of adverse childhood experiences, which is on the x-axis, on a global neurocognitive ability in the y-axis. If you look at people with a family risk of mental illness, which is in orange, we find that as the adverse childhood experiences keep going up, the neurocognitive ability goes down. But in people who do not have that familial risk, it actually does not go down, indicating that there's a combined effect of familial risk with essentially a gene-environment interaction leading to changes in cognition in these people who are unaffected members of these families. Looking more into genetics with what we have done so far, so obviously this is a known figure, so we are looking at essentially two types of analysis, the rare variant analysis and the common variant analysis. With the rare variant analysis, we first started just looking at the first 8 families, and we found 42 rare damaging variants. 20% of them were implicated in Mendelian conditions. Essentially, these are the same kind of genes which are implicated in the Mendelian conditions, but the variant is slightly different. Now, this is a large family of around 20 plus individuals affected with bipolar disorder. Here we found specific targets. These targets were important for us because we're also trying to do stem cell biology and try and understand how these gene variations lead to changes in protein, changes in structure and function. And so here we find 2 specific genes related to CUNC4 and CAP2. These are actin-related genes. Going further with a more recent analysis with 75 families, again we find genes related to neurodevelopment. Of course, a lot of burden on synaptic genes in these families. A lot of them are schizophrenia and bipolar disorder patients. Okay, what it did for policy in the institute, at least, was that there were more people referred for genetic sequencing because people understood that, okay, we would find probably something interesting and relevant. So people started referring patients to us who have a strong family history, who have slightly atypical presentations, earlier onset of symptoms, people who are treatment resistant, and this started happening. This has started happening in the last few years, which is good to know. Now, looking at genome-wide association studies, now we know this slide again that a large majority of these studies have been done in the European American population. We're just catching up. So what we did was we established a consortium. This happened around 2020, and then we have been working on this. So we have established a consortium of 48 psychiatrists throughout the country. All these are institutes, universities with academics and a large clinical load with research capacity. So currently we are working on an NIH grant. This is through the Ancestral Population Network, through which I had met Max. Here we are collecting around 20,000 samples. Further on, we are also working on the genomics of major depression. This is again something that we have been doing over the last few years. We started doing some analysis with some of these samples, and as you can see, the dots, I'm sorry for the lack of pointer, the dots represent the Indian samples, and they are essentially clustered together in the South Asian set, which is again good to know because there is always a discussion that there is so much diversity in India. How will you do a genetic study in India? And that has always been a problem. How do you choose the proper controls? This question has been asked even by NIH reviewers. So we try to look at some of these. We try to look at admixture analysis. We find that it's a 2-factor solution, meaning, if you just divide it by north and south, although there is a lot of clustering together, the divergence is not so high. A panel in the middle shows essentially that the genetic distance within the overall Indian sample is similar to what is found in an overall European sample, which means this can be done. There is still diversity as seen in the UMAP figure on the right side. So what we did essentially was we started looking at some of these. So we generated polygenic scores based on a discovery set based in the Europe, which is essentially all the globally available data. This time we used bipolar disorder and OCD. And we tried to apply this as a test set in India. And what we find here, if you look at the figure on the left, is that the p-values are quite strong, especially for the Bayesian methods, indicating that there is portability of polygenic score across Europe and India. And this is again a new thing, which we are very excited with, and we are trying to now that we are expanding the sample, we are hopefully trying to get better signals. This again shows the decile of polygenic scores, and the odds ratios between the highest and the lowest deciles are quite high, around 20, 15 to 20, which is quite good. So the last step of what I'm going to show is how are we then trying to translate some of this work into biology. And here we are using cell models, again something people know, that we try to use. What we try to do is to generate pluripotent stem cells from blood, from lymphocytes, and then try to study them. So this is our cell culture lab, again established only over the last few years. We have generated around 115 iPSCs, the large team of individuals in Bangalore. We work with the Basic Science Institute, which is a few kilometers away from the clinical institute. And of course, we know that these cells can be then converted to various cell types in the brain. So we can convert cells into neurons, astrocytes, and oligodendrocytes. These are all in 2D. This shows calcium signals in neurons. And as the neurons grow older, the calcium signals actually get better, as you see in panel B. These are oligodendrocytes showing myelin in green, and these are cultured in a bed of neurons so that they myelinate, and you can see, again, I cannot show it, but yeah, you can see a line of neuron which is actually getting myelinated by an oligodendrocyte. Quite exciting to see in the dish. This is all in 2D, but then we are also doing 3D culture. There are a lot of 3D culture work happening in many labs in the world. So we are able to culture 3D organoids till day 125. So you can see that B1 shows the neural tubes. Again, C1, D, all showing the neural lumen, and the synaptic markers are shown in C2. Myelination is shown in E. Essentially, these are all very interesting stuff happening. This can be used to understand what these genes are doing. This can be used to understand what the drugs are doing, because those are very important. So we started looking at one of the families. So this is a family which has many people affected with bipolar disorder. There are gene variants identified which actually had a role in cell migration. So we thought, okay, and these people had MRI abnormalities as well. So we thought, let's look at cell migration in these cells. So that is what we tried to do. This is a representative image, just showing that the cells here, this is what we call an IBD dish. In the IBD dish, essentially, you culture cells on both sides, and the cells have to move towards each other. You can see that the control lines are moving towards each other. The patient lines are moving very fast, but they're not moving towards each other, which is quantified further. So essentially, seeing that the velocity of migration in panel C is higher for the patient lines, but the effective distance was lower, which again, we quantified further with better methods. We tried to do what we call a mean square displacement method, essentially trying to understand whether the cells are moving in a random manner or they're moving in a directed manner. And we find that the patient cells are moving in random Brownian movement manner. Again, we are trying to understand why this is happening. It's still a lot of it is work in progress. Looking at the structure of cells, these are oligodendrocytes which are myelinating. So panel A and panel B, all of them, they actually show the structure or the morphological characterization of these cells. And we find that if you see the patient line, you will find that it is less arborized. And that's again quantified in panel B. The x-axis showing the distance from the cell body and the number of intersections on the y-axis. So the black and blue show the control lines, which actually have a lot more intersections per distance from the cell body. Again, these are organoids showing the bipolar patient lines and controls, and these are what we call neuroepithelial buds that you see, and these neuroepithelial buds actually are lower in the patient lines than the control lines. We also see later that there's abnormal positioning of neuron. This is probably a function of the abnormal migration, which again we are trying to understand. So this is about disease. The other thing that we are very interested to do is to understand the mechanisms of drugs. And here we are trying to understand the mechanisms of lithium, which is a drug used in bipolar disorder. So herein, we are trying to understand mitochondrial biology. And in panel B, we are trying to understand the mitochondrial potential differences between patient and control lines. We are trying to understand whether there is increased cell proliferation, increased cell death. But more importantly, we are trying to understand whether lithium will ameliorate some of these changes. So what we find is that lithium seems to ameliorate the cell death phenotype in these cell lines, and this is specific for a lithium responder, which again is interesting to understand because if there are specific differences between responders and non-responders. Again, lithium at varying concentrations will affect the mitochondria, and I'll just skip the slide. Another student of mine is trying to understand and model this using all publicly available Biobank data, and what he is trying to understand is the difference between lithium responders and non-responders in astrocytes. We got many new targets from this modeling, including a lot of the lipid models, inositol, cholesterol, fatty acid, and these are all not very well known for lithium. So it's very interesting, and now we are trying to validate it in the models that we have with astrocytes. With that, I will stop, and I will thank all my funders, my students, all my mentors, and the conference. Thank you very much.
Thank you, Dr. Viswanath, for that very interesting talk. I'd like now to introduce our second speaker, Dr. Paul Thompson, who is a professor in the Keck School of Medicine at the University of Southern California. His team's research projects focus on neuroscience, mathematics, computer science, software engineering, and clinical aspects of neuroimaging and brain mapping. Dr. Thompson.
morning, everyone. Thanks so much for the invitation and for all of the organizers for creating such an inspiring conference. I appreciate it very much. So I'm going to do a bit of a guided tour of studies across the world, cohort studies across the world that relate genetics to brain diseases. For those of you who are collecting phenotype data, medical imaging is kind of interesting. There's a lot of medical images being collected in clinics every day. Images of structure, function, molecular content, metabolism, physiology, activation. But we don't always see this related to genomics and omics, so I'm going to tell you a little bit about how those two data types are put together. So since 2009, we've run ENIGMA, it's an acronym, Enhancing Neuroimaging and Genetics Through Multi-Site Analysis. We've published the largest neuroimaging studies of over 20 brain disorders as a 45-country consortium. And so the idea is to analyze over 200,000 brain scans, divide the scans into patients with different disorders, and this has led to the largest MRI studies of Parkinson's, epilepsy, ataxia, brain injury. We heard Biju talk about a range of neuropsychiatric illnesses, bipolar, schizophrenia, PTSD, and many others. And not just to look at how they look in a medical image, but to look at the genomic risk factors and other risk factors for these disorders. So this is where the data is. The data is being collected on scanners all over the world. 200,000 scans with MRI. They're not centralized. I mean, they sit in the hospital where they were collected. And the colors are the diseases that that person at that site is studying. So the key at the bottom is a whole range of different neurological, psychiatric, and developmental conditions. So we'll talk about what is ENIGMA, how the neuroscientists and geneticists work across 45 countries. A key to this, as you all know, is diverse leadership in terms of culture, gender, and age. And I'll summarize some of the global genetic studies, how the genetic variants affect the brain, how this depends on ancestry, and also some of the global studies of brain disease in India, Pakistan, Thailand, Japan, and China. So when people are analyzing all this medical imaging data, the first question is how the disorders affect the brain, looking at MRIs, diffusion tensor images, functional images, EEG and MEG. But then also, as we're geneticists, how does genetic variation affect the brain? So you can imagine screening just like any other trait, medical images for effects of common variants, methylation, rare variants, to find genetic and environmental factors that affect the brain's development and aging, disease risk, or treatment response. So this consortium is organized into groups that look at different brain disorders in neurology, psychiatry, and development. You'll see OCD, autism, ADHD, Tourette syndrome, and they're collecting genetic data and brain scans for these conditions. The non-clinical groups are creating harmonized analytic protocols for common variants, rare variants, methylation, even proteomics, and to relate those to the changes that are being seen in the brain. So that's sort of a map of ENIGMA and the different clinical and technical groups that are in it. One question might be, how does progressive methylation of the genome impact changes you see in brain scans? Sylvain de Rivière, who leads ENIGMA's epigenetics group, has found that methylation of HOX or homeobox genes affects the brain trajectory throughout life. And a lot of AI scientists are taking the multimodal data from not just imaging, but also different multiomics and real-time sources to make predictions about diagnosis, prognosis, treatment response, and so forth. So this is how ENIGMA works. If you're a geneticist, this is very familiar to you working as a consortium. People compute measures from their medical images. They test associations between the brain images and clinical risk factors, modulators, treatments. And then there's a huge meta-analysis. And so in different countries, maybe they don't share the data with a central site, but they will tell you the answer from their study, and all of these votes are aggregated. So this has led to, in now 200,000 people, pictures of different brain diseases. So you'll see on the top left there, major depression, bipolar disorder, schizophrenia, alcohol use. These are patterns of gray matter loss in these different conditions. On the right, there's obsessive compulsive disorder, ADHD, autism, epilepsy, and an important neurogenetic syndrome, 22q11 deletion syndrome. And you can even get a t-shirt with this data if you want. So just to show what people do at each site, they collect medical images. Many medical centers around the world are collecting PET and MRI. They process them with standardized workflows, measure the thickness of the cortex, for example. If you come back and have a scan every year, your brain does this. It shrinks unfortunately. So as you come back every year, you can have like a time-lapse movie made. And essentially, all of the repeat scans of these patients are indexed in a database so that you can track the trajectory of gray matter loss over time. So these are an aggregate of patients with Alzheimer's. You'll see gray matter starting in the temporal lobes where the brain's main memory system is, flooding into the frontal cortex, skipping over the sensory motor cortex. And you'll basically see in different diseases, this is a map of schizophrenia, patients with schizophrenia scanned multiple times, and again, the gray matter loss is plotted. You could go back to the biobank and say, what about all the patients that took olanzapine, a newer antipsychotic? What about all the patients that took an older antipsychotic, haloperidol? And you could start to imagine, just like in genetics, you can stratify the medical imaging data in much the same way. Now, here's a trick question. What disease is this? This is gray matter loss, and what do you think this is? It's a trick question. So that is aging. That is normal aging. So we all lose gray matter as we age. This is a cool paper in the Lancet Digital Health of brain measures throughout life. X-axis is age. It's not looking good. The brain's losing a lot of tissue. So you could make brain charts for different countries, or for different cultures, or for different genetic haplotype groups. These are measures for different brain regions, their volume. You could look at function. You could look at molecular content. And you could say, let's use them just in the same way as pediatricians use growth charts. So you know if you have a child, you go to a pediatrician, you look at the height or weight of the child, where are they in terms of their centile versus everyone else. You can start to look at where that person is situated versus all the other people of similar demographics. You could look if they have relapse or remission. If it's someone with an eating disorder, for example, maybe they're recovering, maybe they're deteriorating. But you have to get the basic data to be appropriate to the population that you're looking at. And so an aggregate of all the data across all the world is maybe not going to be the best thing to do. So one of the things ENIGMA has started to do in each country is this group in Thailand studying HIV has been developing brain charts for infants who are being treated for maternal HIV. So they have antiretroviral treatment. Does their brain chart match all the other healthy kids, or is it recovering? Biju, our last speaker, talked about the projects at NIMHANS in India. They are now funded by NIH to collect a sample of healthy elderly folks in India where some of the adverse factors for aging might include access to healthcare, not if you're near NIMHANS, which is a great hospital, but if you're out in a rural environment, it would be trickier. But there's also beneficial aspects in India. The social context, the social environment for elderly people tends to be in many ways better than in countries such as the United States. So you can begin to study and tease apart the different components there. Some of these studies are collecting medical scans in rural environments. This one in Pakistan is collecting data in Peshawar. To do that, you need scanners that work in low-resource environments. This is a little MRI scanner. Zulmurali in Nairobi has been using this to collect data. I would add, if you're at a clinic that doesn't normally collect MRI, you can use one of these. It's designed for low-resource settings, but you can certainly buy one and add it onto your phenotyping that you normally would do. So going back to genomics, those of you familiar with endophenotypes and biomarkers know that you can look at the genomics of any trait, including ones measured in a brain scan. The endophenotype hypothesis used to be that you would need fewer people in your study if you were looking at a molecular biomarker. But Nick Martin, who's a famous Australian skeptic of biomarkers, said if you want to discover genes that affect the brain, you will need images from 50,000 people, just because images are more expensive to collect than measures of height or weight. It doesn't change the power calculations. So ENIGMA has been screening the genome, first of all, for common variants that affect these brain lifespan trajectories. You can do the usual thing. You can measure something in a medical scan. You can do genome-wide association and merge data. So in ENIGMA's genetic studies, people compute measures from medical images. They do GWAS. They meta-analyze their GWAS. And the results look like this. I mean, you can say this is, the volume or the activity of a certain part of the brain. Does each part of the brain have a similar genetic readout? And how much variance is explained by common variants? So even looking at different measures in brain scans, we're now beginning to see that in European ancestry people, up to 10% of the population variance in these measures and scans is explained from just common variants. If you look in African ancestry folks or people from, for example, East Asia, a European discovery GWAS, the PRS from that will only account for about half of the variance as it does in Europeans, obviously motivating much more data collection in non-European cohorts. But you could use this and ask some interesting questions. You say, well, what about the speed of brain loss? Remember those movies of time-lapse changes? Are there any loci in the genome that predict fast loss or slow growth or fast growth? So you could go to the database and say, give me all the people who've had 2 scans at least a year apart. We'll search their genome for polymorphisms that predict the speed, in this case, of brain aging. And you can imagine this could be generalized to other pathologies that progress. And I mean, this certainly works. There's a partnership between ENIGMA and Decode Genetics in Iceland to look at essentially making a library of rare genetic variants and their brain effects. I mean, you could make a long list of copy number variants, duplications, and deletions that have adverse health effects and ask who's had a brain scan, and compile all the brain scans from those folks and see whether the diseases that those copy number variants put you at risk for resemble the patterns of brain differences in the scans. And so there is a very major NIMH effort to compile essentially rare deletion and duplication maps of the brain. And as you can imagine, as the biobanks deepen in content, we can see how generalizable these patterns might be across ancestry. And you can cluster them, just like you can cluster transcripts and transcriptomics. You can cluster brain patterns and see if the patterns of diseases aggregate and whether they segregate with certain deletions that put you at risk for those conditions. Now, what happens when you do this in different countries? So Dr. Viswanath a moment ago talked about the large sample of neuroimaging, clinical, genomic, and biofluid data being collected in Bangalore, India. If you define a normative model of brain aging, and some of the goals might be to understand risk factors for dementia or mental illness, you could try and ask, is it similar to what's found elsewhere? Are there any major differences? So the India ENIGMA initiative that is actually run out of NIMHANS is looking at not just genomic, but also social and environmental determinants of health, which might include literacy or access to healthcare or social support. Multilingualism is interesting. So ENIGMA language is a branch of ENIGMA that looks at whether being multilingual is protective to the brain. It could be. And they also do cross-language benchmarking of neuropsych tests. If you are doing a neuropsych test in a country where obviously English isn't the first language, it doesn't make any sense at all to do that test in English. And so the semantic equivalence of tests is undertaken by that group. A lot of unmeasured health factors are beginning to be measured by these biobanks. So this is Dubai, and it looks like the pollution is pretty high in Dubai. And so this can either be measured through geocoding. We heard Jeff Ginsburg talk about geocoding of data to infer from geospatial maps what the pollution density is. Or you could measure it directly. And so ENIGMA Environment is a working group that will help you geocode your scans and also help you sample things. So if you're interested in radon, which is a big issue in the United States and affects brain development, you can measure it in your cohort. The other question is whether ancestry modulates the effect of major risk genes. So a quarter of us in this room carry the ApoE4 haplotype, which triples your lifetime risk of Alzheimer's disease. But it does depend on your ancestry. So if you're Asian, particularly Japanese, the odds of developing Alzheimer's is much, much higher if you carry ApoE4. If you're European, like me, it's medium. If you're of African or African-American ancestry, the effect of ApoE4 is comparatively low. And in recent years, it's been noticed that although ApoE is the same across populations, this profound difference in lifetime risk for Alzheimer's must be attributable to something. So one of the questions that has been looked at is if someone has admixed ancestry, they might have different ancestry parents, maybe an Asian parent, maybe an African parent. Does the local context of the risk gene and their local ancestral background from the mother or the father impact the overall eventual effect on their life? And it does. It's been found that in the case of Alzheimer's genetics, the origin of your copy of ApoE4, if you carry it, will either make it a very serious risk for Alzheimer's disease. For example, if it comes from a mother who is Japanese, from Singapore, from China. If it comes from an African ancestry parent, that is a much lesser impact. And I won't go into the full story of this, but it's a fascinating view of the modulatory effects of ancestry. Now, we looked in all ENIGMA's brain data as well, and we found that a quarter of us, again, carry this ApoE4 genotype. If you're Asian, it has a very, very high risk for Alzheimer's disease. If you're European, it triples your risk. If you're of African ancestry, it's lower than 3 times the risk. It actually erases about 0.4 standard deviations of your brain tissue per decade if you carry this. So versus people that don't carry it, you'll see this sort of Z-score or Z-score where you have hippocampal volume loss. And this is people that are not ill. It's an ongoing lifetime effect of carrying E4 that varies by ancestry. The other thing that you could look at is look at all the brain diseases. So if you look at schizophrenia, bipolar, depression, if you look at substance use, do the effects of these disorders vary by country? So let's say you scan people in these locations, you ask the people to make a brain map of the disease, and then you start comparing them. You could do a forest plot. So those of you that do multi-site GWAS know that you could look at a SNP and see in each cohort how much it affects the trait. But you could do this with brains. You could say, in these cohorts, I think, Max Lam, who is here in the IMH cohort from Singapore, is one of the schizophrenia cohorts in here. You can say, in every country, does the brain difference agree? Are there any outliers? Are there any people that don't fit this pattern? And one of the things that you see is that to first order, all of the major disorders of the brain in psychiatry and neurology and development seem to affect the brain in a very similar way. And so if you were to make a library of these brain disorders, and here's a number of them in different countries, and this was undertaken by a consortium in Japan called Kokoro. They have 30 sites in Japan scanning people with these diseases. And they've published a series of papers where they've lined up the brain maps of schizophrenia, bipolar, and depression between Europeans and Japanese people. And they found that with just astonishing accuracy, the rank order of brain deficits is identical. In fact, these pictures from Japanese data and European ancestry data are almost exactly the same in terms of the signatures of the different diseases. From China, there's a little bit of a different story. So in China, they also scanned people with major psychiatric and neurological illnesses. This is a partnership between the ZIB consortium in China with 41 cohorts across China looking at the same illnesses. They had enough data to resolve different progression subtypes of schizophrenia. So we looked at how gray matter loss spreads in the brain in schizophrenia. They identified a frontal first subtype, a temporal lobe first subtype. This was replicated in ENIGMA. But they found that the frontal lobe first subtype was more prominent in Chinese ancestry people, whereas the temporal lobe first subtype was more prominent in European ancestry people, which sort of changes the picture a little bit. If to first order things look the same, and then to second order the subtype prevalence may be different, that leads into some very interesting genetic questions. You could do genetics in the subtypes rather than the disease aggregated, and the agreement in genetics of the subtypes may be higher than if you put everything together. Just to wrap up, some lessons learned in ENIGMA working internationally. This is a neuroimaging genetics consortium. It just uses brains as the phenotype. But many of the same things that you all see in your everyday work in genomics hold true. It's politically beneficial when projects are led from different countries. Brain images are very large. They're like whole genomes, so you don't want to be shipping them around. You kind of prefer it if people analyze the data and are custodians of their own data. So as well as this distributed computing, there's been a lot more expertise bringing to bear on these different questions. So just to summarize, ENIGMA is a consortium that coordinates international neuroimaging studies of 33 brain disorders in 45 countries. That has been facilitated by having consensus phenotyping workflows for brain MRI. We didn't talk a whole lot about functional imaging, but it works for that too. And also, just as we would normally do for GWAS and methylation and rare variant calling, agreement on how that should be done. The brain trajectories of the major neurological and psychiatric diseases agree to first order in East Asian, Southeast Asian, and Central European ancestry cohorts, but the subtype prevalence varies. And so many of you who are familiar with dementia know that vascular dementia is more prevalent in certain parts of the world. Alzheimer's dementia, amyloid and tau might be more prevalent in other parts of the world. Loci have been discovered for the speed of brain tissue loss, and there's a brain CNV bank. Those of you studying rare variants can look up what those variants are doing to the brain, if you wish. But we also need to think how local ancestry might modify the major risk genes' effects. And this has been discovered for the ApoE haplotype, which is a risk factor for late onset dementia. Geneticists don't know at the moment whether local ancestry modulates the impact of other major gene effects on the brain. And obviously all of the biobanks that you are pioneering and contributing to will help us discover how and why local ancestry might matter. And just finally, as many speakers have emphasized, the diverse leadership from all regions of the world brings us all new perspectives, as well as discoveries and insights. And so thanks to everyone who scanned people for ENIGMA, a lot of people whose time and effort has been invested in that. And again, to all of the patients and participants in the studies. If you want to take part, you can go look up ENIGMA. It's named after an ENIGMA machine. For those of you that are interested in cryptography, an ENIGMA machine is an encryption machine. So that's kind of why it was called that, as an effort to decode some of the brain and genetic determinants of disease. Thank you very much.
Thank you, Dr. Thompson. That was amazing. Incredible pictures and images. So our last speaker for this session, now we're moving to Latin America, is Dr. Andres Moreno-Estrada, who is a Mexican population geneticist working on human genetic diversity and its implication in population history and medical genomics. He's a principal investigator at the Advanced Genomics unit at the Universidad Institute in Mexico. His work integrates genomics, evolution, and precision medicine in projects involving large collections of understudied populations, in particular from the Americas and the Pacific. Dr. Moreno-Estrada. Before you start, I just want to remind the audience to submit or upvote your questions in the app. Thank you.
All right, thank you very much for the introduction, Gabriela. It's a pleasure to be here. I really want to thank the organizers for this invitation. My first time in Singapore, and it's been so far really fantastic. So yeah, really excited to learn all these exciting projects in this conference. So let me start by briefly introducing my father, who sadly passed away recently a few months ago, earlier this year. He was an anthropologist, and here we have him doing field work in rural communities in Mexico, down there in Brazil, across Latin America. And there I am also in the middle of that crowded truck with my mom and my dad as a little kid in these communities, you know, through all my infancy, without knowing that a few decades later, I would be interested in approaching those communities from a very different perspective, by doing genetic research and establishing my own research lab in Mexico to understand their origins, their genetic diversity, and their genetic risk to disease. So to do that, one of the first things we learn, and one of the biggest challenges we're facing, is that those communities are not represented currently in the vast, you know, majority of databases of genomic data so far, as has been pointed out several times. But I want to stress the fact that this is getting even worse over time, despite the fact that several new cohorts and studies are actually taking place in the Global South. But because proportionally, the other efforts that happen in the Global North are bigger in scale than the actual number of participants of other regions, such as Latino or self-described Latin Americans, are actually smaller proportionally compared to the scale of other efforts. So this picture is, you know, has some mixed interpretations, because even though it's good that we have in absolute numbers new countries represented and new projects going on, we need to scale them up to really enhance the discovery potential that all these projects can have. So that's why we have to not only, it's a matter of representation because we want to be equally represented as other regions. This is also important because scientifically it's really an imperative that these populations have their own characterization, because we have learned over different studies before in the past by us and others that, for example, in Latin America, there's a high differentiation patterns that make them very different even within the region. And this is just an answer-specific PCA plot that is showing only the indigenous fraction of different Latin American individuals across different countries here highlighted in different colors. But you can see that the PCA space they occupy in this plot is even non-overlapping in some cases if you compare individuals from Mesoamerica in Mexico and, for example, the Andean region in Peru or in Chile. So again, the genetic profiling of these populations, even within the same region that we call them all Latin Americans, is really genetically different. So justifying the need of local efforts, which is what we have been doing in the past 10 or so years, trying to understand regionally and launching efforts to do the genetic characterization of many of these populations. And these are just a few examples of that. But then also, we cannot do it locally, just with the local capacity or effort that we have been doing in our countries. We need to scale this up or doing internationally with some endorsement that recently has been actually published in a series of recommendations that the International Common Disease Alliance published a couple of years ago. And there's a series of recommendations how the international community envisions that genetics research should focus priorities in the next years. And one of the recommendations is actually to increase the population diversity of biobanks, and at the same time to promote global equity while doing so. So to respond to that recommendation, we actually formed a Latin American Alliance for Genomic Diversity that we call Latin Genomes for short, and it's a collaborative network of growing members of researchers and countries that has a vision to try to contribute to this challenge, but also keeping local leadership. So our initial goals, and these are the examples that I will be talking about, are to increase the diversity of existing biobanks, as I already mentioned, and also translating the genetic mapping and the genetic diversity to the level of understanding mechanism. In this case, to do a single-cell RNA profiling in different tissues across Latin American populations, and third, by doing so, to increase the local capacity building in the region. So in the first example, I will mention some cohorts that are available already existing in the region that have the potential to be explored and scaled up. And I will mention the Mexican Biobank. There are other resources in the region, such as the Chile Genomic project in Chile, or Poblar, which has a similar aim in Argentina. You can see there different scales, but in general, with the idea of characterizing at the nationwide level their genetic diversity. So in the case of the Mexican Biobank project, which is the one that I'm leading in Mexico, I want to mention this is a nationwide cohort of 40,000 participants coming from the National Health Survey, conducted in Mexico regularly. And the first phase of the project, it characterizes a little bit more than 6,000 samples that have been genotyped using SNP microarrays. And what we have in this collection, apart from this nationwide coverage, is very interesting that we have both a representation from rural and urban communities. It's really representative of the nationwide diversity. It also has associated phenotypic data, not only from the survey, but also biochemical measures from serology measurements that we did. And interestingly, the first thing that we found is, of course, that the whole cohort is highly substructured in terms of having different ancestors contributing in different regions. You can have in the north higher proportions of European ancestry compared to the states on the south that have more indigenous, but we also have other sources, interestingly, African ancestry and Asian ancestry as the third and fourth roots that compose this Mexican diversity. Also in the ENIGMA, we can see that these are gradient, and with some places of the country having highly differentiated patterns, such as the Yucatan Peninsula with the Mayan region. The other thing we can do, because we have such a large data set, is we can actually recover or reconstruct the historical trajectories in terms of population expansions and contractions of different regions of the country that actually mimic the demographic expansions that are known for the pre-Hispanic civilizations in each region. Comparing here, for example, central Mexico and Oaxaca region in the south, they have opposite trends that again coincide with the rise and collapse of different civilizations. The other thing we explore very quickly is, of course, we want to know the number of variants in different functional categories we can find across the genome, depending on the local ancestry. For example, if the individual is of a given proportion of indigenous, European, or West African ancestry, actually that matters in terms of how many SNPs you can find in any of the different functional categories. So again, ancestry at a local level matters. And the other thing we were able to do, because we have phenotypes available, is to replicate some of the known hits for different metabolic traits, such as cholesterol, triglycerides, for example. And what we were importantly able to demonstrate is that when we compared, for example, the Mexican Biobank database with other publicly available bigger resources such as the UK Biobank, and used that GWAS statistics as a prediction for then calculating polygenic scores to then predict the variation in different clinical traits, we found that actually the Mexico Biobank makes an equal or better prediction as the UK Biobank. And you would say, well, maybe this is obvious because you are actually trying to predict the trajectories in clinical traits in the Mexican population. Well, yes, but let's remember that here we are talking about a cohort of 6,000 individuals compared to 500,000 individuals that have the UK Biobank, which has a greater power. So if this was kind of a soccer match, we would say that 6,000 Mexicans beat 500,000 British for predicting clinical traits in Mexicans. So this was a good proof of concept to worth scaling up this resource because it has a good predictive value. And the other thing that we were interested in looking at is, well, let's try to take into account genetic and non-genetic factors to try to predict which are playing a role in dictating the variation in these clinical traits. And I want to focus on one example, BMI. You can see there in the lower left, where one genomic architecture factor, which is the proportion of the genome in ROH, which is Ronson homozygosity, which is also correlated with how much proportion of the genome it has indigenous ancestry, is inversely correlated with BMI. And this is important because has been a lot of proposals saying that obesity is a big problem in Mexico, public health problem, that probably indigenous ancestry has some genetic predisposition for obesity in Mexico. Well, if there's any correlation of any genetic factor, here we're showing that it's in the opposite direction. Just pointing out that probably or most likely obesity has other environmental factors that need to be taken into account. So this has been published already last year. It actually made it to the cover in Nature, which we were very happy about because I think this puts Mexican efforts in the spotlight internationally, and I really like the figure. This is something that I thought a lot about when proposing the figure, and what I wanted to convey the message in this image is, well, genetic diversity in Mexico is not limited to its borders. It's really contributions that go beyond, like north or south, because genetic diversity is a gradient, and that we have this double helix going, both north and south to kind of show this shared ancestry with, throughout the region. But moving into what we're doing right now in terms of application of the whole database, and this is work done by one of my PhD students, Carmina Barberena, is that we're trying to make all these databases accessible to the community. And we're putting that in a web platform called MexVar that basically integrates all the annotated variants in different databases, such as ClinVar, OMIN, PharmGKB, and so on, and then combine it with our database. But what she's doing is then first calculate local ancestry across all the biobank data, and then try to assign allele frequencies depending on that local ancestry for all the variants that are being annotated in these databases. As an example, you have a plot here that shows all the variants have significantly different patterns in terms of ancestry. You see in x-axis those variants that have a frequency specifically on the indigenous fraction of the biobank compared to the y-axis that has that same SNP, but then what's the frequency on the European fraction of positions in the biobank. So clearly what you can say, if there were no difference whether you are indigenous or European in their genome in Mexico, then everyone will be just, not everyone, every variant will be just, following the diagonal. But then the fact that we see some variants, departing from the diagonal, it's telling us that that specific ancestry means for the distribution of that variant. As an example, I want to mention the one that is one of the extremes in the higher frequency indigenous fraction, and this is a SNP that actually modifies the metabolism of fentanyl, which is used in surgery, as you know, anesthetic. So these decreases the metabolism, and you can see if you are agnostic to local ancestry, you would see that the variant is more frequent in the southern part of Mexico compared to the north. But if you look at the local pattern, you would see that it's like prevalent in the whole country, because this particular variant is almost all the carriers out of indigenous ancestry in that particular position. So you kind of want to know your patient has that particular variant in indigenous background, and not just guessing that from self-reported ancestry or just because geographic location, because you would get it wrong. And probably the dosage of fentanyl in the surgery might not be as adequate as it could be. We have other examples. I would just go more quickly on these ones, like COVID risk variants that also have very distinct profiles depending on which ancestor you look at. This particular one in chromosome 6 has a much more enrichment of indigenous ancestry in that particular locus. You can see across all the chromosome. We can have the opposite scenarios when we look at, for example, variants of risk variants for Alzheimer's disease, and this is very in line to what we heard in our last talk, where here we have a very different pattern depending on which local ancestry we're looking at, and the protective allele in this case of Alzheimer's disease has clearly a European origin across the Mexican biobank. So this is also, again, just stressing the importance of local ancestry patterns. So like I mentioned, all this is being integrated in a web-based portal for people to explore and to mine all the database of the biobank, which we hope to release very soon. But like I mentioned, this is all based on, so far, SNP array data. So we are also moving into other partnerships, and the idea is to make the most of this biobank, and we're doing also some genome-wide sequencing. At least a subset of these biobank samples have already been sequenced in partnership with the Genome Asia Consortium based here in Singapore, which is also one of the other reasons that I'm very happy to be here and really get together with this collaboration, together with other samples that have been collected from other regions in South America. So we are trying to really increase the characterization of all these populations at the genome-wide level. But moving on to the different layer that I wanted to talk about is not only genetic variation, but how this variation is impacted gene expression levels. I want to talk about the Human Cell Map Latin American Diversity Project funded by the CCI organization, which is integrating different institutions across different 6 or 7 countries in Latin America with the idea of building a cell map of immune cells across different ancestries in Latin America, and also a new bio-network that is not existing in the HCA catalog so far, which is gallbladder tissue. This is important because particularly in Chile, there's a high correlation between gallbladder cancer and the proportion of Mapuche, which is an indigenous group in the central part of Chile, and these people have a higher risk of having this cancer. So we believe that providing a reference for how the tissue is looking in the healthy population would be very valuable for future studies. And again, while doing this, building local capacity. So we have a common pipeline in terms of experimental approach for either for PBMC tissues or frozen gallbladder tissue that goes first genotyping to ascertain ancestry. We are giving priority precisely to understand what is the ancestry background of participants, followed by the library construction, and then do single-cell sequencing on all these populations. So a key element of all this effort is that we're keeping all the phases of the project locally, from data collection, sample processing. We have 3 different hubs based in Mexico, Chile, and Brazil to do all the data production locally, and then, of course, jointly analyzing the data. Of course, this involves a huge task of community engagement. Going out there to those communities that we want to capture their diversity is a mixture of a strategy between urban locations and isolated locations where we want to really include them in the study. And this is thanks to previous collaboration we have with them in other studies that I have shown before, like the Mexican Biobank and these other community-based diversity projects, and extending this to understand now the gene expression profile. So here we are, the different teams in the field, like in Mexico, in Peru, in Chile, and so on. So to approach those communities, one challenge is how do you go and take a blood sample, which is what you need to be, to capturing the transcriptome, and not only just like DNA, and then you can just keep them, even if it's, from a saliva kit, it can be in even room temperature. This needs to be kept frozen immediately, and then you have a big trajectory between the sample collection and the lab, then the gene expression profile can be modified. So we implemented a new protocol that allows us to go and bring the lab to the community without the necessity of any electricity. No need of centrifuge. We are building this protocol based on a magnetic field that allows us to isolate PBMCs on site, and then this is really, allowing the inclusion of isolated communities that otherwise wouldn't be represented. So the steps basically are, this implies taking or setting up a molecular biology lab in different locations, like could be in the middle of the jungle or the highlands, and then once we have samples, we store them in these containers, snap frozen, and then back to the lab, which is, we believe, the ideal scenario. So just to show you that it's actually working, we have some preliminary data from a pilot set. These are samples from Mexico showing just their IPCA to demonstrate that it's actually of indigenous, highly indigenous ancestry. And we are pooling samples to make the most of it in terms of putting together several samples in a single reaction. We're then demultiplexing them, and here is just to show that we are actually having a good resolution in terms of number of genes, of cells, and number of reads per sample. And on the right, you can see the demultiplexing methodology that we're following that actually are enabling us to distinguish which sample belongs to which individual. Just as a preliminary result, this is very exciting that we're actually finding already some cell types that have a differentially spread profile. Comparing this is just an example between Mexico and Colombia. And ultimately, what we want to understand or get at is genetic variations, since we also have the genetic information that are underlying these these gene expression profiles changes in these different ancestries. So eventually to do some QTL mapping as well based on both information, genomic and transcriptomic. So that's where we are at. I just want to stress the fact that by doing all this project, we're also building local capacity through the local infrastructure. We are setting up all these machines locally, generating the data locally, also with the community engagement, explaining, returning results to communities, keep them informed, and of course, training a lot of students and young researchers along the way, really enhancing the capacity that Latin America can have to lead these kind of projects. I think the ultimate goal is to contribute to this global picture, which we can see that still most of the resources in terms of big biobanks are concentrated in the global north, and there are some promising efforts in the global south. I think we need to really increase these efforts and support them more to try to have a more balanced picture of the world. And I would end just by saying that in this setting, I think I couldn't agree more with what Jeffrey was saying yesterday in the Q&A session, from the All of Us project, when he was asked, well, how to implement biobanks in LMIC countries, and I think he said, if I remember correctly, well, try to seek for international funding, don't work isolated, and join forces. Sometimes starting small is kind of good. So I couldn't agree more in the sense that I think we have been doing all that. So in terms of starting small, I think we have done that already, and all these different efforts have demonstrated that we have been doing the small scale of the job, but we need to scale it up. And we will not be able to scale it up if we don't join the forces, and we don't get the matching scale of support. And also, as it was mentioned in the plenary session this morning, many of our countries, unfortunately, do not have genomics as a priority in their agenda. So I think we really need to join forces and seek those international support. And I really hope that agencies such as the NIH and Wellcome can hear us. And if not, hopefully this platform can serve to raise our voices and really, join our efforts together to, show that Latin America has really a big promise, such as, just like other successful projects did it in Africa, such as H3 Africa. Why not Latin America? I think the moment is ripe for Latin America right now. So with that, I'll just thank everyone involved in these different projects I have mentioned, the Latin Cells Project, Latin Genomes, Mexican Biobank, and the funders, obviously, yourself for your attention. Thank you very much.
So thank you to all the speakers for these fascinating talks and all the work that you're doing in different parts of the world. So we're receiving a lot of questions from the audience. I'll pick this one that I'm sure that is a concern to most of us in the audience, so probably for Biju and Paul. What factors or interventions globally show a positive impact on brain aging or cognitive resilience, considering both genetic and environmental influences?
Is that for me?
I think
Biju, you know this area better than I do.
Talking about cognitive aging, so I think that's your...
Okay, I'll have a go. So luckily there was a Lancet paper on this that came out 3 weeks ago, and it listed all of the factors that promote or adversely affect aging. And some of the ones that are universal, and this won't be a surprise to people here, are cardiovascular factors. So obviously vascular disease in the brain is proceeding more rapidly in people that have poor heart health. And a lot of the cohort studies, as you know, began as cardiac studies and have evolved to collect brain scans and so forth. So it seems almost universally obesity, BMI, cardiovascular health, even statin use, but many of you have been talking about hypercholesterolemia, keeping control of your lipids. The other one that's sort of interesting where we could learn from different cultures is social support. So I come from the United States, even though I sound like I'm British. I lived in the US for a long time. And the social support for elderly people is not good. And if you go to other parts of the world, or if you're from other parts of the world, the social support is much better. And so some of the comorbid disorders of aging, depression, psychiatric issues, can be very, very profound. And I think that's a message that people can learn from other cultures, the ways that elderly people are taken care of and what is done.
Thank you. Biju, if you have insights
I think
to
there
share.
are too many factors. I think there's a lot of work going on around accelerated aging and how it is impacted in various psychiatric conditions. So I think it still needs a lot of work. I think Paul has put up many aspects here, but I think the influence of genetics itself on accelerated aging is not studied much, and that is again something that we need to do.
Definitely. Thank you. So another question, probably addressed to Andres, but I think relevant for all working with understudied populations. How did you get the indigenous communities to agree to contribute their genetic data, and what are your thoughts around that?
No, it's a great question. Thank you for that. So I think I mentioned very quickly, but the success of this community engagement really resides in the fact that we are partnering with a lot of local leaders that already have a long history of working with these communities in the past. Maybe not with the current technology that we're approaching them right now, but it's like just taking one step further from what we have done with them already. So, for example, back in the days, they were already doing some health surveys with them, and then technologies were arising, and then the next proposal, well, now we can use this genetic information to link it with the information we already have from your community to try to understand better things. It's true that in many cases, they expect some quick response to their needs, which is heartbreaking, because many of these communities are really having a lot of struggles with other things that genetics will never help immediately, right? So this is a big struggle. But I think the more we engage with them, the more we can explain that this is like a long-term benefit. And if they are not included, or if they're not participating in these studies, they will be left out, like 10, 20 years from now, when some discoveries can really be applied to that genetic profile that they belong to. Because if they are not participating right now, we will never discover variants that are actually applicable to those genetic backgrounds. So I think this educative effort it's very time-consuming. I think it's completely worthwhile, because this is the basis of trust with the communities.
Thank you. I don't know if you or Paul, you have any insights from the communities that you are working with and how to build trust?
So a lot of our recruitments happen in the hospital. So there it also depends on the rapport between the patient and the doctor. And generally, people in India do consent to these assessments quite easily. Actually, the consenting rates are quite high. In the community, we also ask them, we are taking blood, we do other investigations. That is something that we also say that will help them in some form. So that helps. Yeah.
So thank you. Another upvoted question. Thank you for sharing this. I was wondering if there's any association between these neurodevelopmental conditions like Fragile X and brain aging.
I'm going to say I don't know the answer,
Okay.
but what a great question. And when we've tried to compile, I know, Gabriela, you work on this, so maybe you have a thought on this one, but for the copy number variants and rare deletions and duplications of childhood, there are relatively few studies, maybe, Gabriela, you know of some, that track folks into aging and look at, Down syndrome is a possible exception because of its relationship to Alzheimer's. But what a great question. I mean, I think as the biobanks develop and track people throughout their life, there might be unforeseen surprises later in life that are informative as well. Gabriela, do you know?
Yeah, so we've been, so Down syndrome is an example of that, and we've been doing some work on 22q deletion, that it's a very high-risk condition for schizophrenia, but as we have been following the adult communities, we're seeing development of movement disorders, Parkinson's disease, and faster decline in cognitive functions as well. So that first hit by the CMV probably accelerates other physiologic processes, we think. Okay. So another of the interesting upvoted questions also, the protection of the brain, I think, has been an interest of the audience. So the role of behavioral intervention, what role does it play in genomic and precision medicine? What are your thoughts?
So I think, at least in OCD and a lot of other conditions, the effects of psychotherapy and its effect on neuroimaging, maybe Paul, you could add something there, but my understanding is that it does impact not just medicine, but even the behavioral interventions actually impact the structure and function of the brain. So I think, yes, there is, but I think it's still limited. And I'm not sure if there are very few prospective studies which, which look at, that's true for even medicines, pharmacology, right? Prospective studies looking at first episode, then following them up after medications or even after therapy. Very few studies, very small sample sizes. So I think that is the issue. A lot more scope there, but maybe Paul can add something there.
Yeah, that's a great question. I would make a distinction between neurology and psychiatry of the disorders that we look at, because if a disease has a molecular basis, like Alzheimer's, plaques, amyloid, tau, presumably a molecular or pharmacological intervention is going to make the most sense for that disorder. But I think, Biju, as you just said, in psychiatry for questions such as anxiety, depression, maybe there are other issues, your standard psychiatric therapy can have a major effect. In ENIGMA, our consortium, we have a group that's focused on meditation. And I didn't know that meditation was used as widely as it is, but for people with chronic pain, if people have chronic pain, and you can't take opiates all the time and keep increasing the dose, there's been a lot of evidence that the brain signatures of chronic pain are drastically remedied in at least a subset of patients by contemplative medicine, as it's called. I think when I visited NIMHANS, there was a very strong group in alternative
doing yoga.
medicine. Yeah,
doing yoga.
Absolutely. So, yeah. What do you think? Okay. Okay.
Thank you. So yes, we probably need more evidence on the role of those interventions. So this is a question that I presume was addressed to Dr. Thompson, but I think it can be addressed by all of the three speakers, because it has to do with collaboration and how do you streamline data when you have multiple groups. So the question reads, what challenges did you face in streamlining data consensus across 45 countries? And in all of your cases, are there plans to include more data modalities such as long-read sequencing in your studies? So I think if we can get the opinion from all three of you, because I think it's pertaining to all your presentations.
No, I know that I'm involved in the 45 countries project, but
but a lot of different.
Sure. No, we face similar challenges in that sense, and it's true that data harmonization it's a big challenge in terms of being able to share data across. But I mean, in the Latin cells project, that is the most recent that we are involved with, we have the advantage of having similar protocols and similar platforms installed in each site so that we generate data that is completely compatible in that case. However, when we start combining with probably additional data that we want to add on top of that, will be a very important thing to try to match those variables and types of data that are compatible with this. So we're trying to concentrate everything, in a standardized ways using RedCap, for example, and everyone is capturing their data in a similar way using similar protocols. So that is helping definitely so far. But we're also trying to combine, like I mentioned in other partnerships, additional data that we can get from the same populations. And this includes, in fact, long-read sequencing. This is one of the things we are interested in providing a reference panel for the region, which I think is also another thing that will help in terms of mapping to the local reference instead of just the human genome reference, precisely to try to find more specific variants that are affecting not only gene expression, but in general other variables that we will be capturing. So yeah, this is a challenge definitely to take into account.
All right, I'll have a go. I mean, many of you that are establishing biobanks are familiar with this issue of harmonizing your data collection and analysis with other existing efforts. I mean, this is something that's important to all of us. In ENIGMA, we have dedicated working groups for data analysis harmonization in different areas. So clinical, neuroimaging, genomics, omics, I mean, many of you are familiar with this. And I think fundamentally, some of these are easier to agree on than others. And so if the device is available worldwide, I mean, let's imagine GWAS or MRI, I mean, there's sufficient consensus in the technology, thankfully, that people collecting that data in Singapore or Los Angeles or in Nairobi will be, for example, in the case of MRI, collecting very similar data. Where we've had the most difficulty, because we study brain diseases, is with medication. And so medication, I mean, it is utter chaos how that data is collected. Even if people are trying to do a good job, finding the information from patient records is very, very difficult. So I think one of the things we're learning at a meeting that's about biobanks like this is the value of collecting patient records and adding those to the data sets that are collected, because historically that has not been very harmonious in terms of its collection, but we saw many speakers earlier in the meeting linking to electronic health records and really finding ways to document, systematize, and compare electronic and medical record data as well.
I think Paul has covered most of it. I think even in India there are multiple sites, and for example, having the same harmonized phenotype assessments is always a challenge because each one is following their own, in a way, their own systems. So I think electronic health records, or the health records, many of them are kept actually paper health records, and you have to go through them and spend time. And these are all very thick files, and that itself is a challenge. But that's a gold mine of data, actually.
Yeah, definitely. Yeah. Thank you. We just have a few more minutes, so I'd like to close with a question again to all three of you. So this probably has to do with sustainability and follow-up of these large cohorts. How do you think this can be done in settings of limited resources? Another difficult challenge.
Well, it's very scary. So I think a year ago, I was not sure whether the cohort will continue, whether we can sustain the biobank. We thankfully got funding for 5 more years. We don't know. I don't have an answer to the question because I'm very scared the funding will stop. Yeah. It's always a problem.
Yeah, it's an issue, big effort.
Huge issue.
Well, this is great. I mean, this is basically, touching on my last take-home message where I'm really convinced that this will not happen unless we really join forces and we are heard from who has the decisive capacity to really support this kind of research I mean, I'm speaking for Latin America, but I think that in our region, with the exception of Brazil, that has a really clear link between the health ministry and the science ministry to support this kind of projects. Other than that, in Latin America, we don't have a single example where really this has been put at the forefront of research in terms of local priorities or national priorities. However, if we join forces, if we show that this is something that is meaningful and has the capacity to really even decrease not only disease burden, but economic burden for treating those diseases, and this has to start from external support that I believe, then maybe our local government will say, well, I don't want to be left out because it's my population. I really want to, or I should be supporting that. Well, yeah, hopefully we can shame them and then say, this is something that you should be supporting locally because we are doing it anyway. So we will be doing it, right? So hopefully, again, international funders can hear the priority that this should have.
I will just add to it. I think the last time we approached, for example, even the Wellcome Trust for funding, we were told that unless you have 100,000 people, you will not be considered. You know, that becomes very difficult. We cannot start with 100,000 people, right? I mean, no way.
Yeah, so I think the conversations in this community are very valuable to move this forward. I don't know, Paul, if you have any last thoughts to close on that issue about the international funding and...
Sure. I mean, some of it's a political question. And I was struck by, in Singapore, the politicians' knowledge of this. So you had a number of speakers that are in government that are phenomenally aware of the value of international medical projects. And that's a really good sign. I mean, I think if each of us in our countries lobby for international projects to be funded, it's a political question primarily, isn't it? But I think the more communities like this meet and have examples of synergized efforts across the world, people will see it as a very, very valuable investment. I mean, rather than solving a problem on your own, leveraging other countries' resources and expertise to bring to bear on the same problems. So.
Well, thank you very much. Thanks to the speakers for this very valuable conversation.